This protocol makes it possible to successfully isolate and culture primary cancer-associated fibroblasts from murine breast cancer to screen novel nanoparticles designed to target the tumor microenvironment.
Primary CAF represent the most reliable and physiological model for in vitro studies of tumor stroma. In this protocol, we describe how to achieve an optimal yield.
To begin, prepare a digestion mix for tumor dissociation. And soak the 3 to 5 mm tumor fragments in the solution after tightly closing the tube. Turn the tube upside down, checking that all the pieces of tumor are at the bottom of the tube toward the cap. Then attach the tube to a mechanical dissociator in the proper housing, and run a dissociation program specifically designed for tough tumors. After the run, detach the tube and place it upside down to incubate the sample at 37 degrees Celsius for 40 minutes with gentle shaking. Then reattach the tube to the dissociator in the proper housing and run the dissociation program for tough tumors twice, ensuring no large tissue pieces remain at the end of the procedure. Filter the sample through a 40-micron cell strainer in a 50-milliliter tube. Then wash the filter with 10 milliliters of RPMI 1640 medium and centrifuge. After centrifugation, resuspend the pellet in PBE buffer composed of PBS, 0.5% BSA, and 2 millimolar EDTA, and count the cells with trypan blue. Prepare the dead cell removal binding buffer by diluting the 20X binding buffer stock solution with sterile double distilled water.